Juliana Alcoforado Diniz. I'm an associate research scientist at the Jackson Laboratory in Farmington, Connecticut, and I work in the Robson Lab. And in the Robson Lab, we are part of two tissue mapping centers, the CAPSEN and the Jackson. And my role is involved in the management of the samples that we received, as well as data generation.
What drew you to the field of senescence research?
So I actually come from a very different background. My PhD and my postdocs, they were in molecular microbiology. However, when I joined the Robson Lab, the SenNet grant had just started. So we were starting to receive samples, and it was a very exciting and a new project. So I really enjoyed working with it. But to be honest, I don't come from a senescent background. But I think everything that we apply here and everything that we learn is very, very good.
What intrigues you about the field of senescence research?
So what I think that is very fascinating in the senescent field is that it seems like every question we answer actually open for more questions. And not only the finding new senescent markers, but also seeing the connection with aging and, for example, all the disease. We work with healthy samples, and we clearly see markers for cancer or other possible disease. So I think it's a very interesting field.
Can you briefly describe the research that you and your team perform?
So in the Robson Lab, as I mentioned, we are part of two TMCs. So the first one is the CAPSEN, in which we study kidney, adipose, pancreas, and placenta in human samples from different age groups. We study with healthy and normal organs. And in the Jackson, we study with the kidney, pancreas, and placenta and heart in mice. So we study with young and old mice from 6 and 24 months old. And in both TMCs, we've been applying a vast range of different assays, going from single cell, single nucleus RNA-seq, but also a lot of different spatial omics assays, such as Visium, Xenium, and also some image-based assays, such as Codex.
Please describe the technologies you are using in your research.
The technologies that we have been using, we can actually study in the tissue directly in sections from FFPE blocks. So we can find some new transcripts using Visium spatial analysis, but also from that we can generate markers, probes for Xenium. So we can, for example, for our pancreas, we've been studying the same slide from same donor. We can do Xenium, so we can see the transcripts, but we can also do it followed by Codex. So it's an antibody-based assay, so we can also see the proteins present in that same region. So I think this is very rich, as we can not only find new markers, but we can also validate these markers according to the different age groups of our donors.
How does your research impact the field of senescent cell study?
So as part of two TMCs, we are actually able to study a lot of different things. But I think one interesting aspect of our study is on the developmental senescence. So we are bringing this different point of view, studying genes that may be essential during early development, but they seem to be detrimental in aging. So another very interesting aspect studying with mice in the Jackson is that we are using our outbred mice. So we bring a possibility to investigate the impact of a diverse genetic background of those mice in senescence. Also, our findings so far in the studying with this so-called normal organs, healthy organs in aging donors, brought some very interesting aspects on markers for disease and cancer. And also here in the Robson Lab, we are part of another consortium, which is the MORPHIC. So it's the molecular phenotype of new alleles in iPSCs. So this is a good option that we can generate new alleles from some of those markers that we are finding in the developmental senescence. And then we can validate our findings also in vitro using cell models.
What would you tell students and junior investigators who are eager to contribute to this research?
So I think my advice for young investigators is always to be open to new opportunities, independent of your background. As I said, I came from a very different background, and I was still able to insert myself in the senescent field. I think there is a lot of different things to learn. There's a lot of different skills that we can implement, but also skills that we can learn. So I think always be prepared and always be open for new possibilities.
What are your thoughts on SenNet as a community and resource?
I believe the SenNet Consortium is a great resource as it gives back to the community not only possible new senescent markers, but also new data. So a lot of the data that we've been generating for these different organs in human and mice is going to be available in the SenNet website, so people can go there and do like further studies. Also new technologies and machine learning tools that we've been implementing, and they are also going to be made available to the research community. And also what I already mentioned of different models of technologies. How can we study in the same tissue, transcripts and proteins? So I think this is a very powerful tool to bring back to the community, not only data, but also new technologies. And another point of view that people were not thinking about, this, that could be senescence, that they could be studying that aspect in the SenNet.
Please share any further insights into SenNet or senescence research.
Another very interesting thing that I think from the SenNet Consortium is the possibilities for collaboration. We collaborate not only within our senescent group that we have in our TMC. We have medical doctors that are part of their clinical teams. We have bioinformaticians that are applying all this machine learning, new technologies. So we have all these meetings, but also we can collaborate with other PIs, other universities, people working with different organs. So we can be sharing technology, sharing tissue blocks. So I think the possibilities of collaboration are endless in a consortium like this.